Hello everyone, I'm Zongyue Cheng from Purdue University. Today I'm excited to share our last advancement in deep brain imaging. The green lens has become the preferred option for invasive optical imaging. However, there are two major limitations. The first is tissue damage caused by the surgery. Our team developed a wire cutting tool that replaces the traditional blind needle used during the surgical procedure. This new tool can precisely remove the tissue occupied by the green lens, creating a flat surface at the base. This significantly reduces the tissue damage in the imaging area by hundreds of micrometers. The second limitation of green lens is the narrow field of view caused by the fourth-order astigmatism. By using our newly designed objective lens, neuroscience users can simply replace their existing objective lens to enjoy an expanded field view by 6 to 8 times, easily accomplishing large volumetric calcium imaging. For more details, please visit tomorrow poster board 18. Thank you.
Hi, thank you for the opportunity. So I'm here to present my work about machine learning algorithms through target mode sensory rehabilitation to enhance neuroplasticity post-stroke. So basically, we enrolled 50 adult patients, 26 from US and 24 in Brazil, from Brazil. And majorly we see a lot of pause machine for all this machine learning. We see a lot of good results, good outcomes in this patient compared with traditional modalities. So we see that this is cost effectiveness, and also it's great in low resource countries as Brazil. So compared with patients in the US, this is also can be really, really important for these patients.
Trundles, Flibberwalk, Block, Disseldorf. Oh, I'm sorry. Did that get lost in translation? It actually didn't, because those words are called Jabberwocky, and they have no meaning whatsoever. But Lost in Translation is an excellent movie about these two complete strangers who form a deep, meaningful connection despite only sharing 11 minutes and 30 seconds of dialogue throughout the entire film. This movie is a prime example of how both language and visual cues, such as body language and eye contact, are important for social interactions. As a social neuroscientist, I want to understand how the brain processes both what we see and what we hear to influence how we act and what we do, how we respond. So come to my poster, board 31, tomorrow to learn how single neurons in the human brain encode meaning from spoken words and visual cues during social behavior. Thank you.
All right. Good morning, everyone. What a pleasure it is to be here with you all today. My name is Emily. I am an epileptologist and also an ethicist currently working with Winston Chung at UCSF. I have a deep interest in the implementation of novel closed-loop neuromodulatory devices in clinical practice, of which RNS in epilepsy is an early example. The specific questions I'm interested in relate to complex shared decision-making, such as how best to counsel patient and caregivers on these cutting-edge devices, how to effectively integrate new types of technical expertise in the forms of device representatives and so forth into clinical encounters, and lastly, how to navigate other potential interests at play from research and industry and patient care. During tomorrow's poster session, I'll be sharing data from our study exploring epilepsy patients and caregiver perspectives towards RNS. There will be preliminary recommendations for clinicians, but I believe our findings have broader relevance on the early uptake of novel neural devices in clinical practice. I hope to see you all there. Thanks so much.
Hello, good morning. My name is Josephine Lee Wong, and I'm a PhD candidate in Dr. Yongsoo Kim's lab at Penn State College of Medicine. I'm excited to share with you this project that I've been working on, which is to create high-resolution reference atlases for the developing mouse brain. Complete with anatomical labels from the Allen Mouse Brain Common Coordinate Framework, our 3D atlases span early postnatal days 4 to 14, a critical period during brain development. And to demonstrate their utility, we applied whole brain imaging and machine learning-based segmentation methods to map both GABAergic and microglial cell densities. Finally, we uploaded all findings and resources into an open access web platform where anyone can explore our atlases and cell type growth charts. I hope this work not only enhances our understanding of normal brain development, but provides groundwork for studying neurodevelopmental disorders. Thank you to the organizers for this opportunity and to you all for listening.
Hello, everyone. My name is Ali Tafreshi. I'm a neurosurgery resident working in Mark Richardson's lab at MGH. Our team is investigating a candidate electrophysiological biomarker for OCD in sensing-enabled deep brain stimulation of the ventral capsule, ventral striatum. Our group published a case report in 2022 demonstrating a characteristic peak in the alpha band, and we showed that alpha power magnitude correlated with OCD symptoms. We sought to validate this in a larger cohort of OCD patients, and we found that 12 OCD patients that were in our study demonstrated a peak in the alpha band, which I have demonstrated here, and we tracked this over time. We found that this peak was suppressed on follow-up as Y-box and MADRS scores improved, and that this relationship was statistically significant. We also found that at-home chronically recorded alpha power significantly correlated with MADRS scores as well. And lastly, with the available data that we had, which was relatively limited, we found that when patients were at home and they pushed a button when they were having a symptom flare-up, that this alpha power went up. So I hope that this can help us in the treatment of this disease. And if you're interested, please see my poster in session 3 tomorrow. Thank you.
Okay, good morning. My name is Daniel Taub. I'm a postdoc in Clifford Wolf's lab at Boston Children's Hospital. Thank you so much to Dr. Nye and the Brain Initiative for their generous support. Pain and the opioid epidemic are destroying our country. And the main reason we haven't been able to adequately treat this is our lack of knowledge of pain circuitry and how it manifests within the central nervous system. So using a wide variety of methods such as connectomics, activity imaging, and circuit manipulation, we've now identified a new circuit stemming from the secondary somatosensory cortex that can govern pain thresholds. And so specifically what we're targeting now is using S2 neuromodulation therapies to hopefully produce a new pain therapeutic that's non-addictive in nature. So thank you so much for having me, and please come to my poster. I'd love to talk to you more.
Hi, my name is Lily Torp, and I'm a PhD student in the Burnt Lab at the University of Washington in Seattle, where we are engineering genetically encoded sensors and actuators for monitoring neural activity in vivo. To do this, we use a combined structure-guided and high-throughput engineering approach that allows us to rapidly and efficiently test these sensors in vitro before investigating them in vivo. This led us to develop MuMass, which is a genetically encoded fluorescent opioid sensor that couples ligand detection at the MuOR to an increase in fluorescence of circularly permutated GFP. MuMass is able to sense endogenous, such as metenkephalin, and exogenous, such as fentanyl opioids. Additionally, we are engineering opsin-based actuators that are able to excite and inhibit neuronal pathways using light. And our work really aims to provide novel and robust tools for the neuroscience community and those looking to understand the underlying mechanisms of reward and substance use disorder. To learn more, please stop by my poster tomorrow during poster session 3. Thank you.
Please join me in congratulating those Scholar Spotlight Award winners, runners-up, and of course everybody who participated in the competition.